just outside of, yeah, just between D and, oh, yeah, and DSBC. That's what. It's the layer of stem cells and neurotransmitter cells. Slash.
Sorry, is this human or mice?
Human.
And what's the difference?
Outer radioglia. So, and layers of the cortex.
Okay. Is this applied to primates too?
Yes. Yeah. So the reason why these different zones are important is because here you'll have your this is your neuroepithelial cell, which gives rise to your ventricular radioglia, your outer radioglia, then you have your intermediate progenitors, which then kind of migrate up to give rise to neurons in your cortical layers. So we've mentioned radioglia. What's the function of radioglia during development? Well, morphology is a big hint. So a lot of radioglia will extend from the ventricle to the field surface of the brain, like the entire width. What might that indicate about its function?
It supports the migration.
Yes. Yeah. So it acts
Form
as
a pathway.
exactly. So it acts as a scaffold, but also it acts as like almost like a ladder where these cells, the progenitor cells, can climb up to where they need to go, and then they finish differentiating and maturing. It's what's very cool about the outer radioglia. They can actually divide while they're here. So let's say here's your outer radioglia, and these are its projections. If it were to divide, it would still remain there. And then it can divide even further, maybe let's say asymmetric division to give rise to, let's say it gave rise to another outer radioglia and then a neuron. So what is the difference between symmetrical and asymmetrical cell division? Because it is very important for maintaining the stem cell pool. What's the difference between symmetrical and
Symmetrical generates the same
two of the same cell?
and asymmetrical generates something else,
Yes.
which like make the neural cells like
Yes,
the number of
exactly.
traces.
Yep. So symmetric division is really important in these kind of earlier stem cells because it maintains the progenitor pool. You don't want to deplete it because if you do, you won't be able to give rise to as many cells. So self-renewal, that's also known as self-renewal. It's really important for stem cells, and as stem cells are also characterized by their ability to undergo asymmetric division, which means it gives rise to one of itself and one of another differentiated cell type. So it's really important for stemness. There's a lot of talk in the field now. So when radioglia are able to do this asymmetric division function, a lot of people think that they've like moved up a hierarchy in aspects. So like radioglia are more associated with growth development, and people now believe that radioglia can become astrocytes and then move up to this asymmetric division. It's a very complex process because a lot of things go into regulating this. what are some things that go into regulating stem cells, nerve stem cells? How are regulating this specifically? Simpler question. Are there any neural stem cells in the adult brain? If so, where would they be? Is the brain capable, an adult brain capable of forming new neurons? Yes. And then what process is that really important for? Okay. Why is the adult an adult brain? Why is it important that we're able to give rise to new neurons?
Oh, hippocampus.
Learning and memory. And so where do adult neural stem cells, where are they housed in the brain, in the adult brain?
My guess is it's posterior hippocampus. I don't know. Oh.
Ventricular, anyone? I'm actually blanking on this too, so you're not alone.
Yeah, that's right. It's the ventricular. Yes. So there are layers of stem cells and progenitor cells that line your ventricles. And typically as you age, those layers deplete. And so actually it's an interesting fact that they're now finding with long COVID is that that layer is depleted for people with long COVID. And so there could be premature aging effects in some of these brains. So if you have TBI or stroke or any sort of neurological disorder, this can exacerbate and induce,
It's also impacted a lot in Alzheimer's. So if you've seen a picture of say this is a section of someone's brain, a normal one, the ventricles would be kind of small. But in an Alzheimer's brain, because of all the atrophy, they're larger.
So we'll mention something interesting about olfactory bulbs. So right now we've talked about this lateral movement of cells. What's it called when the cells need to move along this anterior posterior axis? There's a specific term for it, and it's specifically to the olfactory bulbs. We talked about those interneuron layers and subgranular layers of the bulbs. What's another name for anterior?
Anterior? Rostral.
Yes, yeah. So it's called the rostral migratory stream. Rostral tends to be used more for rodents. So rostral is synonymous with anterior, and caudal is synonymous with posterior, depending on which system you're using.
And this is important because there's a big difference between us and rodents or other animals that are on all fours. So we're vertical and they're not, but this is a way to kind of counteract that difference and make it uniform.
So let's do some contrasting of the mouse and human right now. So what's something interesting about size when it comes to neurulation between the mouse and the human proportionally?
Didn't somebody say last week? Humans have smaller neurons. We just have more. Mice have less but bigger neurons.
So proportionally, when this neural tube is folding, it's actually bigger in the mouse than it is in human. So what about time points? We talked about neurulation, we talked about gastrulation, which is when those three germ layers are formed. About what time does that happen in the human during development? So post-fertilization, around when is this happening? So all of this is happening around week 3, 4, and 5 or so in the mouse. So those are with mice. I think the exact time is 6.25 to 9.25 or whatever. 6 to 9 is fine. You don't have to say the 0.25 during development. Does anyone want to get up and start drawing? So she drew gastrulation, which is where we have just 3 germ layers. Does anyone want to get up and draw the stage before that? And then we'll jump into how to make our perfect mouse model. So what comes before gastrulation? Okay, let's start from the very beginning. What's at the beginning, right? You have an egg and sperm. What happens? All right, so your fertilization, which makes, is that haploid or diploid? Now it's diploid, and that's now a zygote, right? So the zygote will divide and become what?
Is it like
No, haploid is only the egg and the sperm. So haploid is where you don't have 2 copies of the genome, right? So we're now at the diploid. So your haploid stage is your egg and your sperm. Why doesn't someone get up and draw it? We should start practicing drawing. Do you want to get up and draw it? Like just you can draw circles and just label egg, sperm,
You want to get up and draw?
sorry, I don't really know what it's talking.
I'll tell you like what to like you can draw circles if you want to.
If you tell me what to draw,
Okay.
I don't know what I'm drawing.
We'll walk you through the stages, but yes. So like, if you can draw me, let's draw a zygote, right? So we're already at our, it should just be a circle, right? This is our diploid fertilized cell. Alright, so what comes after this, right? So the cell is gonna start to divide, and then we have what stage? Well, we have a stage before that.